Okay. Now I'm going to talk a little bit more detail of doing the searches. It overlaps a little bit with Nima showed earlier, so I hope it's not too repetitive. But this is using the query interface to get to the data that we have. How can the query help you? We get a lot of people here who come just for the prediction tools, but we want to let you know that the actual experimental data is also very helpful, and it can help you get a really good idea of what has already been studied and how those experiments went. So you get a big picture about any particular protein or any particular disease state or your epitope of interest. You can see what everyone else has done with that epitope and what assays it was positive or negative in, or what species and what type of experiment. And then if you're just doing predictions, if that's all your particular role is, you can see once you've predicted a lot of epitopes, if they've been tested and were they tested for how you plan to use them. So you can save a lot of time. If you can see something was very highly tested and always negative, you might not go ahead and make that peptide and do further experiments. You may rule it out right there on the spot. So we use a lot of words we probably already have up till now that are IDB specific words that we need to just refer to our fields, so we have some word to call them. So if I say something that doesn't make sense, stop me and ask me, because you're probably not the only one who doesn't understand what I said. So we talk about the epitope source and source organisms, so those are usually the protein and species that epitope came from. For non-peptidics, it may be the larger carbohydrate or something. And then the assay parts, we have the image and the antigen. For us, it's what the host was originally exposed to that helped trigger the immune response, and then the antigen is what they tested in that experiment. So if it's a tetramer, what peptide was loaded into the MHC. If it was a lysa plate, what was actually on the plate. That was the measured response to. And then we have in vivo and in vitro processes. So for us, an in vivo process is an exposure that happened in the living organism. So you had a disease or you injected an animal with something. And the in vitro is you may have taken, say, naive PBMCs, and then you put them in a plate with overlapping peptides to stimulate them. That was an in vitro process. And then because we use those ontologies, there's relationships between our terms being parent, child, and siblings. So for example, virus family is the parent of influenza virus, and different strains of influenza are siblings with each other. And then this is the link to the curation manual that I mentioned. It's also on the website if you want to see how we curate and more information about the details of the names of anything. And then on this homepage that Nima showed before, there's these little question marks next to a lot of our terms, and they give you more information about each term that we've chosen to put into the website. And we put a lot of thought into them, but if they don't make sense to you, let us know, because that means we should keep thinking about what we should call them. And so on the homepage, this is the most common search interface that most people use when they come to the IDB, and within it are embedded all these finders. And these are the finders take you to the ontology hierarchy trees that you can search. And these are the search panes that Nima showed of the different epitope, antigen, host, assay, MHC, and the disease. And then this is an example of one of those finders opened up, more than what I showed before. So this is MHC, and it shows you that hierarchy of you can search by higher level class or specific level human class 1 or a specific MHC molecule, or you can group them all together, in this case by their serotype. So this is a unique feature that we came up with so that we could use the ontologies. We knew we wanted to do ontologies, but we wanted to make it useful. So we created this interface for each of the ontology backends. And we hope that it helps you search a lot of data more quickly and easily. And it lets you be narrow to specific or high level, depending on what your interest is. And often if you do a query and you don't get a lot of data back, go back to what parameters you chose and pick a higher level, because it may just be there aren't that many studies at that fine level, which would be good for you to know. And these are all the ontologies we're using now, the uniprot reference proteomes, the KEBI for the chemicals, NCBI taxonomy for the epitope source and the host, MRO for the MHC molecules, OB, disease ontology, and gazetteer gives us geolocations. We get countries of where the hosts are from. Sometimes that's relevant for exposures. We also have identifier search. We're not going to demo this today, but we want to point it out. So up at the top, there's a specialized search dropdown that gives you the ability to enter. Does this have? I don't know what it's called. Okay. So if you use that dropdown, you can get to the way to enter just an identifier search. And this lets you enter the epitope ID if you have it, if you want to see there's new data for an epitope you've been studying for a while, or if you want to put in a PubMed ID or a PDB ID or a ChemB ID to get to a specific structure, you can do that here and you bypass all the other search parameters. So if you have a specific ID, it's faster. Then I'm going to do a very short demo query that's going to repeat some things we did before, but with a little bit more detail. So first I'm going to start with the antigen, that this is the protein or species that the epitope's derived from. And what I want to show is that you can type very little if you know synonyms and you don't want to type out human herpes 5. Because we use NCBI and they provide all these synonyms, you can type just CMV and then you choose from what shows up. And this again is just to make it faster and easier. And then I want to add that you can multi-select. So if I then go on to type EBV, then human herpes 4 shows up and I can select from that, and then the little number 1 will show you that you've selected 2 things. And you can keep searching if you want to add more. And then here I'm also adding MHC search, and again, because we're using that ontology and we get the synonyms, instead of typing HLA-A asterisk 02:01, which is a pain, I just typed 0201, and it popped up because of the synonym without having to type as much, and then I can pick it. And the more you use the IDB, the more intuitive that becomes, hopefully. Then, so what I want to show you is that by default, positive assays is always checked. If you want to include negative assays in your experiments, you need to uncheck that. And then it will show you everything you've chosen. And before it will search, you have to hit the search button. And this is because we can't foresee how many different parameters you're going to build. So you can put something in every box, or you could pick something in just one box, and we won't know when you're done. So you need to hit the search button. And then you get the search results, as Nima showed. And I'm going to do a quick overview of what's on each page. So at the top, it shows you all your search parameters, and you can click them off up here. If you left positive on and you decide you want to take it off, you can take it off now. And then each of the tabs has its own set of headers, and Nima went through what each of these is. And then on each tab, it has its own set of column headers, and this is dependent on what type of tab you're on. So epitopes have source antigen, source organism, and you get counts of how many references studied that epitope and how many assays were done. So you really quickly get a picture of how often a particular epitope is studied, and it often is like this, that one is studied a lot, and that's often used as a positive control, and some are very rarely studied, and this usually is because there wasn't a big response and there was no need to keep studying it. But maybe that was in a cat and you're studying mice, so you might want to still look at it. And then on the epitope page, this is where you get the meat of the data. That includes all the links to everything, and these are little filters that you can narrow by anything that you see on the page using these little filters. And then this is the immuno browser that Nima showed you. So when you go to the antigens tab, it's by default you'll get this pop-up. So it'll always remind you that that's the icon for the immuno browser. And when you take that pop-up away, then you get, these are the results for the antigens tab, and I wanted to point out that every one of the columns is sortable. So the little arrow icon, you click on that and it sorts. And because my query had 2 organisms, 2 different species, we have proteins here from both species. So I chose to sort on the species so that I could group together all of the proteins from CMV that we had data for, so then you get an idea about that species. So this particular protein was not very well studied, and then other proteins are much more highly studied. So that also reflects what's known about those proteins and how interesting they were to other people. And then antigens tab, oh yeah, I guess this is the sorted. This is the sorted, these are all the reference proteins for that species. And then on the assays tab, you see that we have sub-tabs for T-cell assays, B-cell assays, MHC assays. They're separate because they have different header types describing, so there isn't the MHC restriction column on the B-cell tab, for example. And again, each of these is sortable, and it's where you get to the meat of the information. And then this is the receptor tab that Nima showed. And you can see all the CDR3s, and if one is blank, it means it wasn't shown to be paired. That doesn't mean it isn't paired in real life. This is all reflective on what sort of data was available in the references that we curated. And then the reference tab that Nima mentioned, this is where you get all of the information about the publications the data came from. And you can click on each one and go to that publication, or you can export the whole list if you want a list of references about what you did your query on. Then one thing, we talked about these results search panes where you can search more things. So each of these has either exactly what was on the home page, or it has additional search parameters once you're on this page and we have more room, so we can add more features. They're just not used as often, and that's why they're not on the home page, to keep that simpler. But we are presented here because some people do want to use them. So I'm going to go through the differences between here and the home page, the additional things that you can do now that you're on the results page. So in the epitope search pane, we have the ability to search for a particular peptide or different matches to that peptide, different identity levels. So if you searched on a specific peptide and you got no results, then you should lower your threshold and search for more different identities, because different authors search slightly different strains, or they may have searched a longer peptide. And then you can also search for discontinuous and non-peptidic epitopes, and you can look for modifications to a peptide. If you're particularly interested in a specific modification, you can select that here. And then also 3D structures. I'll show a few slides about that. And then the antigen search pane is the same as the home page. But the receptor search pane is new. It doesn't exist on the home page. But it lets you search for the sequence type, just anything that has a 3D receptor. I mean, a receptor sequence information, if you want to start there and just see what's known. And if you get a lot of results, then you might narrow it to specific antibody or TCR types, the chain types. And again, we have the identity options, where if you've sequenced a TCR and you want to see, does anyone know what the TCR binds, you can put just the CDR3 in, or the full length, CDR1 or 2, in and search to see if someone else has identified it already. And if you don't get a hit, so you can then lower the threshold again and see if maybe a similar TCR was sequenced. And sometimes you might find that you expected it to bind one thing, but it binds multiple things, or it binds nothing. But this data, we're trying to accumulate it as much as we can, but we're still dependent on what people publish. Then more filters. So the host is the same, the MHC and the host are the same as on the home page, so there's nothing new there. Disease is the same as on the home page. But then we additionally have this reference search pane, and this lets you search for PubMed IDs, for submissions, but also you can put author names. So a lot of people like to put their own name in and see what data we have for them. And then but you can put the title of the paper if you know the specific title, or you can do a date range if you're only interested in new papers or old papers. And then a little bit more detail about each of the type of details. So the epitope details, you get to it by clicking here, and in each of the tabs has the same concept. And it takes you to this page that Nima showed before, but I want to point out how it summarizes all the data we have for that particular epitope. It tells you how often it was tested, if it has 3D structures, how many assays it's been tested in, and it accumulates everything from all the papers that we've curated. I chose one that's very highly studied, so it has nearly 700 T-cell assays. And for each of those contexts, we give you additional information of how often the particular context was studied and how often positive. So this is the MHC section. So you can see AO201 has been studied for this particular epitope a lot, and it was always positive. Some other MHC were very rarely studied and were negative. That doesn't mean they don't necessarily bind, but they're not as exciting to look at. Then this epitope having all those T-cell assays, you can see what types of experiments people have studied for that particular epitope. So not very often was it studied for IL-13, but it was studied for interferon gamma quite a bit. And it was I think, always positive. So that gives you an idea of what type of an epitope this is. It's a summary of this is clearly a class 1 T-cell epitope. And then just a few B-cell assays, but it still gives you some information about what's been done and how often it's been studied. And then now on the assay tab, this is the assay detail column. I think I'm not going to show you the details, but you can click on that ID and it'll take you to the details that I think Nima is going to show later. 3D structures. So we capture all 3D structures that show antibody, TCR, MHC binding to an epitope. And if you want to just see what data we have for that, you can click this in the epitope search pane that has 3D structure, and then your results will show you all cases that have 3D structure. And what you see on the assays tab is this little 3D icon. And any query may retrieve some 3D structures. When you click on that little icon, you open up our epitope viewer, and it gives you the ability to navigate through that structure. Where am I? The receptors page. So like all the other tabs, the receptors tab has the link to IDs. When you click on those links, you get to the details, and so this summarizes all information for that particular CDR3. How often it was studied, what epitopes it bound to. So with that CDR3 paired with this specific combination of CDR1 and 2 in the other chain, was shown to bind to this epitope, but this combination with that CDR3 was shown to bind quite a few different, very similar peptides, so it gives you an idea which residues matter and which residues don't for that interaction. And then there's links to all the information, those epitopes and their assays and other what papers they came from. Then back to the assays tab. Yeah, export. That's what I'm showing. So the export result, I think Nima pointed this out. I'm going to walk through what happens when you click on that. You choose between two export types. The one with IRIs, the IRIs are the links to those external ontology identifiers. So if you want to see the link for that particular term for that assay type or that chemical type or whatever the link out may be for each term, this file will include all of those link outs, where the other one will not. And then when you click it, you get a little pop-up page that tells you where you can access your results and what your query was that you are exporting, because you may want to keep, if you're doing a lot of different queries, you may want to keep this information with the export. And it gives you a link to downloading the full database. We have a lot of other download types. So if you want to see everything, if your query is really large, you might as well take it all and then further narrow. And then I want to show you two things real quick about the assay export. All of our exports have 2-column headers. So I get questions about this that confuses people. The top row of the header is the group of fields. So the reference is a group, the epitope is a group, and the host may be a group. And then the second row is the field within that group, because there's a lot of IDs and it's to provide more context for those types of fields. Then another question we get a lot is why we have 2 columns for proteins and 2 columns for organisms. And this is because of how we organize the data. So every epitope that's protein is assigned to a specific protein that it's 100% conserved in. And that will have a specific identifier, a specific sequence, and a name. And there may be many different names for the same protein from different GenBank records. And we need to access those different GenBank records because it's that specific sequence that was studied. But to group them all and to make it easier for you, we use those reference proteomes. And so the first column is the protein that was specific for that epitope. And the second column is the reference proteome parent for that protein. So there you'll group proteins with different names for the same protein using the reference proteome name. And so if you sorted on this column, you would group all the proteins together that are essentially the same protein. So it's there if you need to sort your data, and then that's the parent accession. You can see that different names have different same accession here and then a different name here. And then the same thing is true for the organisms. Many authors will test in the context of a very specific strain. That doesn't mean that peptide isn't present in other strains. It means in that paper it was tested in that context, and we want to be true to those authors. So the authors gave these epitopes in this particular strain. But for you to organize your data, if you want to see everything that was from human herpes 4, you can search using the parent species. So it's just the species level, where there may be a strain level here. If the authors didn't provide a strain level, then we still have the same generic without a specific strain. This is really what the authors said, and this is the grouping column, and it's the same here. This is 100% identical to the epitope sequence, and this is a grouping column. And some people only care about one of the other columns. That's up to the different user types. And then more exports. Like I said, we can export everything. We're a free resource, so we want you to take us and use us however you want. So there's up at the top of the home page, there's a drop down for database export, and when you go there, you'll see that we have all sorts of different exports. I'm having trouble operating this. So CSV exports, MySQL exports, and I want to just talk real quick about these two that are our newest export types, but you can play with them and export everything. The receptor export is fairly new, and it contains all of the sequence data we have for TCRs and antibodies. So it has the full length sequence or the nucleotide sequence, the CDR1, 2, 3, and the VDJ gene usage, if provided by an author. And it tells you every epitope that every one of those receptors bound to. So it may be multiple epitopes, sometimes by one paper, but maybe 3 different papers report the same CDR3 binding to 3 different, slightly different epitopes. And the 3D full export, this is all 3D data where there's a PDB structure showing 3D structure of the receptor and the epitope. So you can export all of this, and it includes the PDB IDs and the contacts between the receptor and the epitope, and it'll give you the full length receptor and antigen sequences. And if you combine this with the receptor export, you'll be able to combine the CDR3 and the VDJ gene usage with those contact information. I think that's my last slide. Yep. Okay, any questions? Okay, so the question is, how are positive and negative assays defined? So we go by what the paper said. So one author's cutoff, say for an ELISA, it may be, they may set a very high threshold based on whatever their background happened to be in that context. So we don't differ from what the author said. If the author said all the healthy controls were negative, we'll capture that assay as negative because the author said it was. Or sometimes they will be really nice and provide, at this standard deviation cutoff, we consider that positive. But it's based on how the authors decided what was positive. So, I also want to add that with autoimmune disease, it's really common for the authors to set a different cutoff for the different patient groups. So they may say in the RA patients, we consider this negative, but in the lupus patients, we consider this negative. And we don't argue with authors, we represent what they said and believe that they did that for a good reason. And if we can't tell, we will write the authors.
So it looks like, I already answered my question, but your output when you put in, for example, a linear sequence and then the resultant table, is that prioritized by quality of the data, whether it's real positive or low positive, et cetera, or how is that table prioritized?
So if you did, the question is how are the results tables prioritized? So if you did, for example, an epitopes peptide search, the order in which the results come back to you is how frequently it's been studied, not how frequently it was positive. But typically, the more positive ones are more frequently studied. And you can sort by any column.
Just a follow-up question. I don't really use those export links, but if I were to, it seemed like the description of those links was quite extensive. So if I were to scroll over those links, would it provide the description of what that what that output will be, or will I have to do the output and then kind of figure out on my own, oh, it's providing this data and that data, et cetera?
The question was, how do you know what you're going to get in your export? Is that right?
Yes.
Each tab has different column types, so the assay export will be different from the epitope export, but in general, what you see on the screen on the results page gives you an idea what columns will be in the export. It's just the export takes what was shown to you as one column and breaks it out into all the details. Gotcha. So like the immunization column on the assay page will be, like 10 columns in the export where each component is broken out into more details.